Hi all, hope you all are doing great. My name is Chaturya. Today I am going to talk about extraction of DNA from blood kit method. Introduction. Extraction of DNA from blood is a very easy task in laboratory nowadays. The sample of blood is treated with detergents to break open the cell membrane, spilling its contents. Enzymes are now used to break down all the protein, RNA, sugars and fats in the solution. Ethanol is often used in final stages of DNA extraction as under the right conditions, as DNA will dissolve into it, but other components of the cell will not allow the separation of DNA to be used for further analysis. Firstly, let us know what is DNA. DNA is a deoxyribonucleic acid which is found in all living things. It carries the genetic information in the cell. It contains instructions for our body cells to perform their specific functions. The sequence of nucleotides determines the individual hereditary characters. The basic unit of information in DNA is. Now let us see what are the required materials. Gloves, tissue paper, blood sample, 15 ml centrifuge tubes, Eppendorf tubes and EDTA tubes. And the chemicals we need are reagent A, reagent B, tea buffer, sodium perchloride, ice cold chloroform, ice cold ethanol. And equipments we need are centrifuge, Vertox, water bath and rotating mixer. Here in this slide there are only few, but we need a lot more materials. Principle. The isolation of DNA usually begins with the lysis of the cells or tissues in order to destroy the protein structures and allows the release of nucleic acids from the nucleus. For extraction of DNA from blood kit method, there are three basic steps. They are cell lysis, protein precipitation and DNA purification. And firstly, let us talk about RCB lysis. Lysis is nothing but the breakage of cells to remove membrane lipids. And firstly, we need to prepare 3 ml of whole blood sample in EDTA tube. Here we use EDTA tube because it contains heparin which prevent coagulation. Now transfer the blood sample to 15 ml tube and add 12 ml of reagent A. Now mix on rolling for 4 minutes at room temperature. Centrifuge at 3000 rpm for 5 minutes at room temperature. Cell lysis. Here comes the white blood cell lysis. Add 12 ml of reagent B on cell pellet. Vertox briefly to resuspend the cell pellet. Add 250 ml of 5 molar sodium perchlorate. Mix by inverting several times. Place the tube in water bath for 15 to 20 minutes at 65 centigrades. Cool this at room temperature. Protein precipitation is the process in which protein is separated from any extra contaminants that may be mixed with it. Firstly, for protein precipitation, we need to add 2 ml of ice cold chloroform. Now mix on a rotating mixer for 30 to 60 minutes. Centrifuge at 2500 rpm for 5 minutes. Transfer upper phase into a clean 15 ml tube using a sterile pipette. Note: here we need to use a glassware when we use chloroform. DNA precipitation is a process in which the nucleic acid is precipitated using alcohol and salt. Ethanol and isopropanol are the two most common types of alcohols used in DNA precipitation. Firstly, we need to add 2 to 3 ml of ice cold ethanol gently on the tubes of the walls. And now invert gently to allow DNA to precipitate. Centrifuge at 2500 rpm for 3 minutes. Discard the supernatant gently. Allow to air dry the DNA pellet by inverting the tube on a tissue for 10 to 15 minutes. Nucleic acid may need to be determined here. And the next comes DNA hydration. Resuspend the pellet in 100 to 300 ml of tea buffer. Rehydrate DNA by incubating at 65 centigrade for 1 hour and leave it overnight at room temperature. Centrifuge the DNA sample briefly by a short spin. Transfer the DNA sample to 1.5 ml Eppendorf tube. And so here is the flowchart which represents the entire extraction process of DNA. And we'll take the sample and add RBC lysis buffer to make the cells lysed. And then we'll add GB buffer or proteinase K and we'll centrifuge it at 60 degrees centigrade for 20 minutes. Upon centrifugation, it leaves a cell lysate, and that cell lysate is sent into the lysate column. That column and DNA will bind together. That binding mechanism is termed as DNA binding, and then that column is washed with ethanol to get rid of all the other components except DNA. And after the wash step, we'll get only the elute part, and will not get any other cell components. We'll get only the DNA. After elution step, we can finally collect the purified DNA. And this is how we'll extract the DNA from blood kit method, and this is how the whole process takes place. Thank you. And if you like this video, please do like, share and comment. Thank you.